Salt of the Earth Farm Stories is proudly brought to you by Grig Media.
If we keep going the way that we're going and the crayfish follow the trajectories that they're on now, we won't have any Murray crayfish left by the end of this century. No feasible populations.
Today, we're heading into the southern tributaries of the Murray-Darling Basin to meet a true freshwater giant, the Murray Spiny Crayfish. It's the second largest crayfish in the world, covered with an impressive armour and full of surprises. Joining us is Dr. Scott Raymond, who's spent years studying this iconic species. He shares fascinating facts, some key do's and don'ts, and even how to check if a female is carrying eggs without uncurling her tail. We'll also hear about a painful pinch moment and a bit of blood, and his unforgettable encounter with Sir David Attenborough. It's a sharp, surprising and thoroughly interesting chat with the clever and passionate Dr. Scott Raymond. Let's dive into it. Good morning, Scott.
Morning, Darren.
Scott, can you tell me, who do you work for and what's your position?
I'm a freshwater ecologist at the Arthur Rylah Institute in Melbourne.
And the Arthur Rylah Institute, they're a research organisation?
That's correct, yeah. We're the research arm of the Victorian government, so anything to do with crayfish or fish, plants, mammals throughout the state, we go out and undertake research to answer specific questions about the threats, how healthy populations are, impacts from fires, that type of thing.
And can you just list some of the research projects you've taken on over the last 5 to 10 years?
Well, previously I was investigating the use of farm dams for raising threatened fish species so that we could reintroduce them into the wild. Prior to that, we were into river restoration techniques and how that impacted populations. So looking at movement data, when fish move, why they move, how far they move, do they return, those sorts of things. In the last 5 to 6 years, I've really specialised in freshwater crayfish, particularly Murray crayfish. They're a harvested species with a large recreational fishery behind them. So what our focus has been is to look at the impacts from harvest on the health of the population using sex indicators, so differences in sex ratios, changes in the size structure, changes in densities over time and in different regions. So we look at harvested versus non-harvested populations and compare the size structure, sex ratios, that sort of thing.
Now, mate, each time I catch up with you, you're in the best office in the world. It's either maybe on a farm dam or on a quiet little stream somewhere. Things are quiet this morning here. It's beautiful. Can you tell me where we are today and what are you up to?
We're on the Ovens River near Bundalong in northeastern Victoria. So we're researching crayfish, specifically Murray crayfish once again. So the current project is investigating the size at which crayfish carry eggs under different thermal conditions. So the Ovens River, as you know, stretches all the way from Lake Mulwala, which is a large warm water fishery, and we look at the thermal profiles all the way up into the alpine regions and looking at the number of eggs that each of those crayfish hold relative to their size. So from that investigation, we've found that the large crayfish actually don't hold eggs until a much larger size. And because size and the number of eggs held, there's a relationship there. They hold a large number of eggs versus the same species in higher altitudes, which, funny enough, reproduce at a much smaller size.
So are we saying small? How big are they likely to be?
So the ones in the Murray River in the lowland populations on average hold eggs at about 107 millimetres in size, whereas the crayfish from the alpine regions can hold crayfish at a smaller size as 50 millimetres.
They can hold eggs at 50 millimetres.
Yeah, they hold the eggs at 50 millimetres. So what we found is we were trying to investigate whether the harvest slot limit protected all of the crayfish within the thermal profile of a system or systems, because we were counterintuitively thinking that the lowland populations, because they're large and they carry a lot of eggs, that those populations would be more stable than the upland populations. But because the females that are holding eggs in the upland populations have multiple breeding events across multiple years, they're providing offspring to the populations continuously before they're available to be removed for harvest. Whereas in the lowland populations, they're growing so rapidly that they're halfway through the slot limit, meaning that actually half of the female crayfish don't have eggs in the slot limit, so they're available for harvest before they contribute to the following generation.
So do you mean slot limit, that's the harvest period?
That's the harvest slot size limit, which is currently 100 to 120 millimetres carapace length.
Just describe that. That is from, where do you take that
That's
measurement?
from the back of the eye stalk, so the hard section on the cephalothorax, to the rear of that plate, so where it articulates with the tail.
Now I want to get onto the message about how we check if they're females and if they've got eggs or not in a moment. But can we go back to the start? Can you describe the Murray spiny crayfish? They're a prehistoric looking creature.
Yeah, they are very prehistoric looking. So they're the largest decapods in the systems, meaning that they have 10 pairs of walking legs.
That makes a decapod.
That makes a decapod, yep. So they're the largest freshwater invertebrate. Often you'll find that they contribute over 80% of the invertebrate biomass to any river system. So biomass, the weight of organisms within a system is their biomass, and because of their large size, they often contribute to in excess of 80% of the invertebrate biomass within rivers and streams throughout Victoria and elsewhere across the globe.
Right. So what do they eat?
They're characterised as polytrophic, meaning that they're herbivores, they're detritivores, and in some instances they're carnivores. So predominantly they'll consume leaf litter, sticks, but they will consume worms and other small invertebrates. And in the carnivorous sense, they're usually found feeding on dead animals rather than actively hunting live prey.
Right. So they won't eat a little yabby or anything?
They definitely will eat a small yabby. They will eat others of the same species if they can catch them. They're very aggressive.
Are they?
So they separate in size classes throughout river systems. So if you have tributaries, you'll often find smaller crayfish trying to get away from the larger crayfish, and they'll occupy the small interstitial spaces amongst rocks and within snags and try their best to stay away from larger, more aggressive conspecifics, of the same animals, just larger ones.
So it could be quite an aggressive show under the water there sometimes.
It could be. I mean, there's been a few studies to show that within the water that they're less aggressive, but we often see them when we pull them out of pots. So we're removing them from their natural environment. Even though they can move on land, their preference is obviously the aquatic environment. They are so aggressive. Every single opportunity they get to squash fingers, to cut you, to nip you, they will take those opportunities. They've got, it seems as though they have a bit of foresight. They know exactly where you're going to put the ruler, and they're on top trying to nip you constantly. Yeah, they're fun to work with.
Well, in the off-the-wall questions at the end, Scott, I want to ask you about a pinch moment, so we'll get to that in a sec. And they're quite territorial, aren't they?
The males are very territorial. So when there's low population numbers, males will set up territories up to 10 to 20 metres. the size of those territories will be reduced if the densities of females and males are higher, because the females don't have to go as far to find a mate. But yes, they're very, very aggressive.
So when is cray season and what are some of the rules?
So in Victoria, well, it's best to check obviously with the Victorian Fisheries Authority handbook, because it varies slightly depending on whether they're closed water bodies, and some closed water bodies versus other water bodies vary as well. But typically in the river systems, it's from the start of June to the end of August. So you have that 3-month period where you're able to go and capture and remove crayfish. So the regulations, so you have a size limit that we talked about before, this occipital carapace length, which is from 100 to 120 millimeters. You're allowed to capture 2 crayfish within that slot limit per day, and you're allowed to have 4 in your possession at any one time. But you have to maintain those crayfish as a whole animal. You cannot remove the tails or the claws.
And they have to be males?
No, they don't have to be males. So this is a bit of an issue that we've done quite a bit of research on. So often the crayfish regulations explicitly state that you cannot capture and remove a crayfish that has eggs. So there's no explicit determination on the difference between male and female crayfish. So the problem arises when you have female crayfish within the slot limit that don't have eggs. So they're available to harvest. And we know that females in our population models are critical to long-term sustainability of populations. So not only do we have the problem that females within the slot, under certain circumstances, well, pretty much 50% of the females don't carry eggs for only 50% of the time that they occupy that slot limit, but that those females that do have eggs lose eggs per handling event. So overall, typically adult female buried crayfish will have from 300 up to 1,800 eggs. But each time that they're incorrectly handled, they can lose up to 40 of those eggs. And when you consider that these female crayfish over the harvest season of 3 months typically move anywhere from tens to hundreds of meters, they can be caught multiple times and therefore lose multiple eggs over multiple handling events, which is quite concerning.
That has a huge impact on the population.
That has a very significant impact on our long-term population projections.
So how do we check?
So the easiest way to check whether female is carrying eggs is to have a look at their sex organs. So males and females are dimorphic, so you have males with sex organs at different locations to females. So females, if you hold the crayfish, so it's the underneath is facing you, you look down and on the 3rd pair of walking legs, it will have a plate. If that plate...
And that plate is a round plate, about 5 millimeters in size, roughly?
That's right, yeah, exactly. If that plate protrudes, hairs called setae will be around that plate. So the hairs come out as the plate moves forward, and that is an indicator that the female will have eggs, or she will have eggs in the next harvest season. In contrast, the males have papillae on the 5th pair of walking legs, so the bottom pair of walking legs, and they're external protrusions of about 5 to 6 millimeters. So
that's a little 5 millimeter round knob that sticks out?
Yep, exactly.
Right. So if we see a female with a little 5 millimeter disc on her 3rd walking leg with hairs out from underneath it, she's got eggs.
with a high probability of greater than 90% of the time, she will have eggs. There is a small portion of females that will mature in the next year that also have CT that won't carry eggs in their first year, and that proportion is about 10%.
Right, and so for that fisher person out there, they've picked it up, they've checked, it looks like it's a female with eggs. What are they to do?
So the concern for us is people that pick the crayfish up, unfurl their tails, and in that process of unfurling their tails, it initiates a flight-fight response from the female crayfish that vigorously flaps her tail. And that's where we see a lot of egg loss, because the eggs are only semi-adhesive and they're guarded within the tail, but they're very, very loose within that tail. So if she starts to flap because people have intruded and unfurled her tail, she will flap and eggs will just fly everywhere.
So we don't want to uncurl their tail, but we know that she's most likely got eggs. What do? Do we put her straight back where we found her?
Yeah, so the regulations state that you must immediately put that female back into the water. So our advice is to place the female next to the bank, next to some habitat like tree roots or rocks.
And if it's a male and it's between 100 millimetre and 120?
Then you've got to follow the regulations and decide whether it's worth removing a crayfish from its natural habitat and consuming it.
Yeah.
Or whether you put it back.
Actually, I'm just looking over your shoulder at the moment. There's a couple of your colleagues that have just come in. They've been out in the water most of the morning collecting some pots, and this is part of a research program that you've got going on at the moment. They look like they've got a pretty good job as well.
They've got a fantastic job. It's really, really exciting pulling up a pot and finding 20 large crayfish and, you know, emptying them out and trying to band them without getting nipped. And they always scurry into the corners of the boat and back into nets and other nets and pots, and constantly trying to get away from you until you pick them up. Then they're constantly trying to get you.
And I often joke about the office that you're in, but there are other times too where you've got to be inside at a desk putting all this research statistics together.
Yeah, look, this is the most fun part of the job. A lot of the other time we're back talking to modelers and statisticians and biometricians, hydrologists, trying to understand what factors impact on changing population numbers or sizes or sex ratios, that sort of thing. For me, that's a really, really exciting part of the story, because you're getting the data, you're figuring out what the data is showing you, whether it's answering the question that you've asked, which you've designed your research around that question, and then you've gathered the information. For me, having a look what other people have found in other crayfish species and then trying to relate that to what we've found, and even going as far afield to have a look at the impacts of disparate or different sex ratios from moose populations in Canada to sturgeons in North America to eagles to marmots. It's fascinating. I mean, every single study that you do just gives you a little bit more insight into the ecology of multiple animals and how different ecosystems operate. And that's where you start to ask new questions. Oh, does my animal operate like that? How does this population, you know, achieve these findings, whereas this population is markedly different? Is it because it's being harvested? Is it because of altitude, photo periods, predators, food availability, habitat, you know, river size, complexity of habitats? That there's a multitude of questions and answers as just trying to figure out the right question, the right design, and to get the answer that hopefully explains what you've actually found. That's the key to good science.
Is there a key moment in your career that you've uncovered some new information that really stands out to you?
I think for me, probably changing the reproductive indicator from size at the onset of sexual maturity to size at functional reproduction was probably the most impactful for our model and for the species, because it changed the way that not only that we viewed the species, but it changed the trajectory significantly of populations based on this realignment of what reproduction means to us. Because like in humans, size is not a very good correlate of reproductive capacity for the following year. So you can have large female teenagers, for instance, at 13 who have no probability of contributing to next year's generation. So we moved it from size to something closer to perhaps, for example, a 25-year-old pregnant female
Yeah.
who had a much greater probability of contributing offspring to the next generation compared with a younger,
Yeah.
large female.
Yeah. Is there a misconception with crayfish? Like, they've been in this world and in this country for thousands of years. Like, they're not going to go anywhere soon.
Well, they've been virtually unchanged for 350 to 400 million years. So when Pangea split up into Gondwana, there was an aquatic bridge that the species from what is now North America moved into what is now Australia. And they're virtually unchanged. I mean, they're still decapods. They're still showing the same food and eating habits. They reproduce in a very, very similar way. They look the same. There might be different sizes and different colours because of different niches, but largely they are a hugely successful species with, genera with over 600 species dominated by Northern Hemisphere American species and Australian species.
But we've got to look after them, don't we? And there's things that we can do by just checking for that they might have eggs.
Absolutely. I mean, look, this all stems back to commercial harvest in the 1950s that went all the way through to the 1980s, where we saw 50, 60, 70% declines
Oh, really?
in population numbers. Yeah, well, in some years they were removing 15,000 tonnes of crayfish from our waterways, across South Australia, Victoria and New South Wales. So they were quite large numbers, none of which we've ever seen those sorts of numbers since that time. The average size of crayfish has reduced, probably due to harvest and more probably due to selective harvest of large males, because we've had quite a few changes in regulations, beginning from no regulations to you can take crayfish above 80 or 90 mil, and then they've had restrictions on slot limits and numbers and whatnot. But
so this is important, isn't it? To get these messages out there that you go and enjoy them, but be mindful that we could lose them if we keep doing the wrong thing.
absolutely, if we keep going the way that we're going and we follow, or the crayfish follow the trajectories that they're on now, we won't have any crayfish, Murray crayfish, left by the end of this century.
Wow.
No feasible populations.
Wow. How long do they live for generally?
They have a maximum life expectancy of 50 years. So our population models are 50-year age structured stochastic population models. So they take into account lots of variation, but typically you'll have crayfish that start to breed at 70 to 80 mil. I mean, that's temperature and location and size of habitat dependent. So there's quite a bit of variability. So particularly in reproduction. So if they're isolated in large reservoirs that are thermally stratified, they can breed at 6 degrees, whereas in other populations, they'll need temperatures to reduce to below 14 degrees Celsius to initiate breeding. So there's quite a bit of variability.
Is there any future research you think that's needed to help them survive?
From my perspective, so we're all about trying to align conservation with management, because obviously both of those aspects of Murray crayfish are really important. For me, it's more from a conservation angle. My preference at the moment, given we know very little about the species, is to focus a little bit more on reproduction. So the timing, the cues to initiate reproduction, how long mating goes for. We know that the brooding period is about 140 days, but the vast majority of the research has been undertaken decades ago and from selected locations. And we know that the variability in this particular species over large thermal profiles of rivers, so temperature gradients, varies quite a lot. So we want to have a closer look at whether the current regulations are adequately protecting populations across their whole profile, rather than looking at individual characteristics of the species that were recorded from one location 50 years ago. So we're trying to be a little bit more location specific, so that at the moment we are using a one size fits all approach to harvest, where we think that that could be problematic.
Is there one key take-home message you would like to get across to our listeners?
Yeah, have fun, be safe. I mean, they're a beautiful animal to look at and to enjoy. They're ecosystem engineers, they're icon species. They help shape the environments in which they live. So admire them, respect them, put them back if you will. If not, make sure that you adhere to the regulations and look after our girls.
All right, Scott, I got some fun off-the-wall questions here for you, mate. These
Okay.
can be short answers and you don't have to answer them all. If a Murray spiny crayfish was a superhero, what superpower would the females have?
More eggs and internal fertilization.
Nice. All right, here's a good one. I'm looking forward to this one. Have you ever had a crayfish pinch moment during your research that made you rethink your career?
Not rethinking my career, but there's been quite a few pinch moments that I've seen on other people. Recently, I took out a volunteer and I was warning the volunteer how dangerous these claws can be. So to be very, very whack, as I looked up and was explaining it to him, it bit me on the bottom of the wrist. And thank goodness he only got me by a couple of millimeters, but it sliced straight through.
Really?
And I was trying to cut my wrist with my hand and explain to the young fella how dangerous they were while blood was dripping down my arm.
Right.
So it made me look a bit silly, yeah.
If a crayfish could send one message to humans, what do you think they'd say?
Stop trying to rebuild our homes. The system was working fine as it was. Every time you try to rectify or change or modify the system, you're hurting us.
Well said, mate. Is there a crayfish fact you love telling people that always surprises them?
Probably the reproductive cycle of how they release their eggs over the sperm packets that the males deliver near the oviducts. And with the eggs, there's these Glare glands that break down the spermatheca packet. So all the sperm come out. As the sperm comes out with the eggs, they travel into the tail. and inside the tail they have these pleopods.
Mm.
So they're like fine feathers, and they move around all over the place. So you get this washing machine action inside the furled up tail
Yeah.
of a massive sperm and eggs. And once those eggs have been fertilized, there's another gland called the cement gland, and as you can imagine, that produces a semi-adhesive gel which helps to tie each of the eggs onto the hairs on these pleopods.
Ah,
And then you get these setae that wrap themselves around the hairs to
yeah.
enclose the eggs and to try to protect them, even though they're externally
Yeah.
fertilized and held. And then they're covered with this huge armored spiked tail. So yeah, that's probably my favorite part of
Yeah.
the reproductive cycle of crayfish.
Fascinating.
There's so many aspects of their life history and diet and evolution and speciation and niche differentiation between altitudes and river systems and water clarity, water quality, flows. Everything seems to impact on how they grow, how they reproduce, and how healthy the populations are. So there's a lot of factors, and unfortunately, when we do muck around with the systems, and you have pollution, and then you have people not following the fisheries harvest regulations, it disrupts those natural processes, and the only ones that suffer are the crayfish.
Crayfish. Is there a surprising reaction that you've ever had from somebody after you've explained your career or what you do for a living?
Probably 30% of the people have a wry little smile and ask me, yeah, but do they taste good?
Scott, what do people misunderstand about you most?
Wow, jeez. Probably how much excitement an animal that's rarely seen provides for your career and just your health as a human being. I love being out on the rivers. It's calming. It's provides an opportunity for 3 or 4 of us researchers to sit around at night and talk about what we found during the day. What does it mean? What's an area that we don't understand? How we could potentially look at answering these multitude of questions that just seem to arise. You answer one question and it opens another 10 doors. And through each of those doors, you have the whole funding cycle and the permit cycle, and then where do you go to undertake that research? I mean, it's a magnificent job. I mean, being involved in the outdoors and scientific research is just the cream of the cream for me.
Because you often go out for a week at a time and you've got a camp on the edge of the river and you're living it, aren't you, really?
Yeah, well, one of the regulations, the permitting regulations from fisheries, is that we have to be within eyeshot of the pots,
Yep.
just to ensure that other people, don't muck around with the crayfish that we capture. So, yes, often, well, most times we'll go out for a week at a time because we have a specific sampling protocol and design that we use. So it's comparable between systems, and so space and time. So we've got obviously replication in each of the sites. So we'll have site replication within site replication, and we'll follow that through over the years so that we're confident that the changes that we see in the populations are real changes. So we don't get specific errors because we're adjusting the way that we're sampling the crayfish, for argument's sake. But yeah, we live out here for a week, often 2 weeks at a time. It's just beautiful. You get the whole experience, even if the cockies up here keep you awake most nights. It's beautiful. The birds, the sight, the sunsets and the sunrises.
Well, mate, talking about birds, just a couple of minutes ago when we were chatting, there was my favourite bird ever, the azure kingfisher was just over your shoulder on a log, and that was a special moment.
They're beautiful. They are such little characters. They will sit there for 10, 15 minutes, bobbing up and down, and all of a sudden, bang, they'll
Fishing.
be into the water. They'll pull out a little smelt. And as you're going along the river, setting nets, they'll often, it looks like you're pushing them forwards. So each time you come up to encroach to get a better look, they'll fly another 20 feet.
I had a career highlight here with you last year, actually in the Barmah Forest.
That was fantastic.
And we crept up within just a couple of metres from an azure kingfisher, and it let me get it on camera, and it was smiling beautifully.
They are magnificent birds. They're just beautiful.
All right, mate, back to the off-the-wall questions. If you could have a coffee or a cold beer with somebody, who would you choose? And they could be dead or alive.
David Attenborough, without a doubt.
Oh, gold. I'll sit in the background and listen to you two talk.
I actually met him.
Oh, really?
Yeah.
Seriously?
Yeah, a mate and I were in Borneo, and I really needed to go to the toilet. And we came to this resort, really flash resort, beautiful place. And I went up to the guard, so they had an armed guard there. And I said to him, mate, I really need to go to the toilet. And he's like, you can't come in here. And I said, well, it's better I go in than right here. And he's like, yep, go. So we went, mate went up, got a couple of beers. You know, it's always hot over there. I did what I had to do, came back, and my mate was making this funny head movement, you know, like look over my shoulder, and I said, what's wrong with you? And he whispered, David Attenborough's on the next table. And I was like, no way, let's go. And he's like, oh, you can't just walk over there. And I said, well, just watch me. And I walked over, and as I walked over, he looked up, and I said, oh, g'day, Dave, how you going? You know, my name's Scott. I said, huge fan, mate, you know, love your work. I said, my friend and I are fellow biologists, and thanks for the 30 years of inspiration from the BBC, you know, to everything that you've done since. And he's like, oh, yeah, would you like to have a seat?
You're joking?
No. We were like, oh, this is magnificent. And my friend said, oh, look, you know, would you mind if I took a couple of photos? And he was sitting down with the script writer and his photographer. And the photographer was like, oh, hang on. If you're going to take a photo with the man, let me snap a couple of photos for you. And he actually said, oh, I was a bit surprised. Well, I was hopeful, and he did. He sent all the photos through. He grabbed our cameras and took a couple of photos. So we've got these arm-in-arm photos with us and David Attenborough.
Mate, that is absolute gold. Well, mate, we'll wrap up this episode. Your colleagues have just come in from upstream there with a boatload of crays, and you've had a big day. You're out early at 7 o'clock this morning, I believe, collecting pots. So, Scott, a privilege to talk to you, mate, and thanks so much for your time.
Oh, mate, absolute pleasure as always. Thanks heaps, Darren.
A big thanks to Scott for sharing his knowledge, stories, and field trips, from egg checks to pinch moments. Please follow us on your favourite podcast app, and keep your hands dirty and your spirits high.